guys, welcome to our YouTube channel. Myself Upasona Panday. I'm a first Prof Medicos from NILRATAN SIRCAR Medical College and Hospital. In this video, I have discussed about the mechanism of the transmission of the impulse and the properties of the conduction of the impulse. And with this video, I am concluding this chapter and I will be taking up a new chapter in the next video. So stay tuned, watch the full video and join our Instagram and Telegram from the given links. Okay, now watch the video. We were in our discussion in the transmission of the impulse, and so far we have discussed about the synapse, that is the structure of the synapse, the neurotransmitters and the types of the synapse. And today I was supposed to start with the transmission of the impulse from one neuron to the other across the synapse. So this is our familiar picture of the synapse with the presynaptic part and the postsynaptic part. Now this is the axon terminal. You see this portion, and the waves of the depolarization has reached the terminal part of the axon. So it will now reach in the synaptic knob. You see these red lines showing the waves of the depolarization that has entered the synaptic knob. Now these waves of depolarization will cause the voltage sensitive calcium channels to open. The voltage sensitive calcium channels open up when the wave of depolarization reaches the synaptic knob. Now this Ca2 plus from the extracellular fluid will rush inside the cell, inside the synaptic knob, due to the opening of the voltage gated calcium channels. These Ca2 plus channels ions along with some proteins, the V snare protein, the T snare protein, they interact together and cause the fusion of these synaptic vesicles with the presynaptic membrane. You see these synaptic vesicles have fused with the presynaptic membrane, where now it has fused in the active zones of the presynaptic membrane, the thickened portions that are known as the active zones. At those regions, the synaptic vesicles have fused. Now further, this calcium causes the exocytosis of the synaptic vesicles, and these neurotransmitters are released in the synaptic cleft. So let us recall once more this: the wave of depolarization reaches the synaptic knob, then this wave of depolarization causes opening of the voltage sensitive calcium channels, which causes the entry of the calcium ions from the ECF to inside the cell. And inside the cell, that's inside the synaptic knob, these Ca2 plus channels will interact with some other proteins. Those are the V snare protein, the T snare protein, and will togetherly cause the fusion of these synaptic vesicles in the active zones of the presynaptic membrane. And also these calcium ions causes the exocytosis of the synaptic vesicles, which ultimately releases the neurotransmitter in the synaptic cleft. Now the neurotransmitters have been released in the synaptic cleft. It will go towards the postsynaptic membrane. There are the receptors. Those are approximated towards the active zones by the cell adhesion molecules, and those receptors will catch the neurotransmitters. You see this, I have shown a few neurotransmitters being catched by the receptors, and when these are catched by the receptors, there is the transfer of the impulse, the transmission of the impulse from the presynaptic neuron to the postsynaptic neuron in an uninterrupted way. So this is the mechanism of transmission of the impulse. Look at this chart. Depolarization reaches the synaptic knob, then the voltage sensitive calcium 2 plus Ca2 plus channels open. Calcium fuses into the synaptic knob from the ECF. Then interaction of proteins occurred along with the calcium, which ultimately causes the fusion of the synaptic vesicles with the presynaptic membrane, Then this calcium causes the exocytosis of the synaptic vesicles, and ultimately the neurotransmitters are released. This is all about the mechanism. Okay, are we clear with that? Now, there are some properties. We have so far discussed everything about the generation and the conduction of the nerve impulse inside a single neuron, or the transfer of the impulse from one neuron to the other neuron. But this is a very intricate process, and this has some particular characteristics. The first one being the most important is the all or the none principle. I forgot to write it down, but just listen to it and write it by yourself. The all or the none principle. In this, what happens when an action potential is generated? When an action potential is generated, the impulse will be transferred, the impulse will be transmitted throughout the neural system in an undiminished way. Once it has been initiated, it will do its work. It will carry the impulse to the central nervous system without any failure. If the stimulus isn't enough, then the action potential will not be initiated. Once initiated, it will carry the impulse to the central nervous system in an undiminished way. There comes the importance of the threshold potential, the firing potential, minus 55 millivolt. If this potential is reached, if the stimulus is strong enough to fire the axonal membrane into minus 55 millivolt of potential, then the action potential will be generated. Once generated, the action potential, the impulse will be carried throughout the neural system in an undiminished way, undiminished and uninterrupted way. That is the all or the none principle. We have done with that. Now what we'll look? Now we'll look into the one-way other characteristics. That is, one is the one-way conduction of the impulse, the synaptic delay, the synaptic fatigue. What is this one-way conduction of impulse? In this one-way conduction of impulse, when we talk about the structure of the neuron, we always state that the dendrites carry the impulse towards the cell body, and the axon carry the impulse away from the cell body. So it has a particular direction for the flow of the impulse. Why is it so? Because at the synapse, when the impulse is transmitted from one neuron to the other neuron, then at the synapse, the neurotransmitters which carry the impulse are only present in the presynaptic membrane. So it can only carry them to the postsynaptic membrane, and in postsynaptic membrane there is no presence of the neurotransmitter, so the back way is not possible. That is why there is a one-way conduction of the impulse. Now the synaptic delay, what is this? Now when these neurotransmitters are released from the presynaptic membrane, then it reaches the postsynaptic membrane, and there it generates action potential in the next neuron. It needs some time. That time is known as the synaptic delay. That is basically the time difference, the time that is required by the neurotransmitter to travel from the presynaptic membrane to the postsynaptic membrane and create a wave of depolarization there in the next neuron. That is synaptic delay. Now let us see this synaptic fatigue. What is this? When suppose one wave of depolarization has reached, that will cause the fusion of the synaptic vesicles, and then the neurotransmitter will be released. It will reach the postsynaptic membrane, and the action potential will be generated in the next neuron. Immediately after that, one more wave reaches there. So few more neurotransmitter will be associated in that process. Again, immediately one more. And if this continues for some times, then what will happen? Now one time will come when all the neurotransmitters available at the moment get exhausted, get used up. And till new neurotransmitters are made available, there is no way for the conduction of the impulse from one neuron to the other. At that time, the condition reached is known as the synaptic fatigue. It means that due to the exhaustion of all the neurotransmitters present, there is the failure in the conduction of the impulse from the one neuron to the other neuron. That is the synaptic delay. Are we clear with these concepts? Okay. Now here is one information punch for you. That is, When the neurotransmitter is the acetylcholine, then the acetylcholinesterase enzyme present in the postsynaptic membrane breaks the acetylcholine into acetate and choline. Why? Because what is the function of the neurotransmitter? Its function is to carry the impulse. So when it is done with its function, it should be reused by the body. For that function, it is broken into acetate and choline, that is the usable forms, so that it can be used in the formation of more acetylcholine or maybe some other component of the body, right? Now we have to discuss the solitary conduction of impulse. How many types of nerve fibers we have talked about? We have talked about two types of nerve fiber. One is the myelinated one and the other is the non-myelinated one. This is the myelinated one and this is the non-myelinated one. Okay? What is present in the myelinated one? Now in the myelinated one, the Schwann cells around the axon grows concentric layers of the lipid sheaths forming the myelin sheath. So you see, these are the myelin sheaths, and in between the myelin sheaths there are gaps. Those gaps are known as the node of the Ranvier. So these are the myelin sheaths and these are the nodes of the Ranvier, and the impulse jumps from this node of Ranvier to the other. Why this is so? Because the presence of the concentric sheaths of the lipid make the portions of the axon non-conducting. Those insulates those portions of the axon. The extracellular fluid is present surrounding the axon. The extracellular fluid contains the ions which makes the fluid conducting in nature, and the ion channels are present throughout the axon, but these are more concentrated in case of the node of the Ranvier. So the ions can easily pass to the extracellular fluid and the vice versa can occur, making these regions conducting, whereas the lipid sheaths insulates the portions of the myelin sheaths, and there is no passage of the ions from or to the extracellular fluid. So those portions of the axon cannot conduct the impulse. So the impulse jumps from the node of the Ranvier to the other. This is known as the solitary conduction of impulse in case of the myelinated one. But you see, in case of the non-myelinated one, there is no myelin sheath. So the impulse has to pass all through the nerve fiber. Okay, so this is the basic difference. So the solitary conduction is a faster method of conduction and it should be mostly present in the central nervous system, whereas this non-myelinated one should be present in case of peripheral nervous system. So we are done with the discussion of this generation and conduction and transmission of the impulse. Thank you for watching the video. Stay updated, and from the next day I will be taking up a new chapter and will be concluding the chapter very soon. So stay tuned. I will be taking up digestive system and will discuss the concepts of the chapter and then go to some questions. Okay, and in this chapter we are only left with the reflex, eyes and ears. I may be posting the video later, but for now I will be starting with the digestive system. So if you like the video, like, share, comment and subscribe, and take care. Bye bye.